Welcome to the Honeybee Watch Symposia series. Hello from all over the world. I'm Steve Rogenstein, the project director for Honeybee Watch and your host today. And people probably know Arrigo by now, our science director. He is behind the scenes. He's our master of technical issues and resolving them, and also the moderator of the Q&A that will be at the end of today's program. So today we'll be hearing from 3 PhD students who are at different stages of their studies, and they come from different universities and different countries, but each of them specializes in studying free-living honeybees. And today we're going to be hearing about their areas of focus, what observations they've made so far, any current data and findings that they can share, and more, hopefully anecdotes too. And once we've heard from them individually, I'll moderate a panel discussion between the 3 of them. And then at the end of the program, Arrigo will lead the question and answer session with the audience. So just to start, I'll briefly introduce Honeybee Watch for those of you who are unfamiliar. We are an international coalition tasked with better understanding how free-living honeybees survive via natural selection with little to no human intervention. And by free living, what we mean are bees that have chosen where they want to live, whether that's in a tree cavity, in a log hive, in a hole in the ground, or in human-made structures such as building walls, chimneys, or even abandoned beehives. And our aim is to find nature-based solutions to lower mortality rates of managed colonies, to increase beekeeping's sustainability, and ultimately to protect any threatened wild populations around the world. In our 5+ years, our research partners and citizen scientists have identified more than 2,500 nest sites on 5 continents, and that number keeps rising. One project that we have been working on is coming to its conclusion. After 2 years, a task force that we put together to reassess the red list status of Apis mellifera in Europe, a collaboration with IUCN, the International Union for the Conservation of Nature. We have some game-changing recommendations that will be announced next month, so stay tuned. It's very exciting for us after all this work that we've done on it. We're also part of a consortium that applied for and was awarded a 1.1 million euro grant from Biodiversa Plus and the European Commission. And that's to extensively study free-living colonies in France, Ireland, Poland, Portugal, and Sweden. And that's a project called FreeBee, which you can learn more about at free-bee or free-bee.eu, as Arrigo just dropped into the chat. And our primary mission of forming this global coalition, one of the kind of initiatives that we embarked on 3 years ago was to start the Symposia series. And it's to connect the community, including you, to one another. So now you kind of have the basic skeleton of what Honeybee Watch is all about. And now I'm going to contextualize what the Symposia series is about, especially for our newcomers. First of all, we have something like 390 people who have signed up for today's event. So super exciting. I think that that's the best attendance we've had so far. So thank you for your support. So we launched a series 3 years ago as a means to bring together the free-living honeybee community. And it was an opportunity to link scientists and experts from around the world so that they could facilitate the exchange of expertise, data, best practices, so that you, the audience, could be introduced to their work and you could link to one another. And obviously, we're committed to exploring and discussing challenging topics that we have faced and other researchers in this community have also faced. So in the first year, we explored monitoring protocols, for instance, which resulted in an open source document that's free to download from our resources web page, which Arrigo already put in there. And there, like I said, you can get all of our other videos, but also there are articles on free-living bees, natural beekeeping, tree beekeeping. There are videos, there are journal articles, there are posters. So explore that resources page if you want to get more information about these topics. In the first year, we also discussed data sharing and data rights and how to define wild in the context of free living colonies, which is a difficult challenge because wild can be construed in so many different ways when related to bees. In our second year, we explore bee lining, tree cavities, whether wild honeybees are wild bees too, and best practices for communicating and recruiting for citizen science projects. And then this year, we kicked off in March with an introduction to Free Bee. That's that study that I referred to before from Biodiversity Plus. In June, we talked about free living honeybees in Africa. And then in December, to round out the year, we're going to be exploring bee-related policy issues. And if you want more information about the series or to register for December or for the past so that you get the links to the videos, you can sign up on our events page, which Rigo just dropped into the chat. So that brings us to today's program, which we're calling the latest buzz, current research from Honeybee Watch Partners. We're going to go to our third guest, Kaylin Kleckner, who came to us through Jamie Ellis, her PhD advisor, who's been fascinated by free living colonies in South Africa and wanted to study them more. And when we learned about her work, we reached out to even know more about it because, as some of you know already, Honeybee Watch has a commitment to expanding into Africa in meaningful ways. So, of course, we couldn't pass on the opportunity to learn more about her research and explore any opportunities to collaborate. And as you'll hear today, her work with Apis mellifera scutellata and capensis is groundbreaking, with really informative data and observations. And we're excited to see where she potentially goes with this after her PhD studies in Florida.
Thank you for having me. Um, this today is going to be a snapshot of my PhD research. As Alex has already shared, there's a lot that can happen in these 4 or 5 years. And so today I'm specifically going to focus on my work with Ness Natural History of Wild Honeybees in South Africa. So the Western honeybee, the whole focus of the seminar today, has an extremely large native range that spans across Europe, the Middle East, and Africa. We've heard a great deal about Apis mellifera in Europe so far, but there's an extensive portion of this range with many diverse subspecies of honeybees that is relatively understudied. In Africa, their honeybees are, of course, an important pollinator, but they also fuel the beekeeping industry in quite a different way. At least learning beekeeping in North America, where honeybees are not native, if I were to want to start beekeeping, I could call a beekeeping supply company and even order a package of bees in the mail. And from my understanding, in some other developed areas, like in Europe, where there's a commercial, intense, managed population of bees, it can look quite different than those free living colonies. But in many parts of Africa, beekeeping is produced by capturing wild or free living swarms in catch boxes. So, a robust wild population of honeybees is required for the success of beekeeping endeavors, whether that's beekeeping with nature based hives, top bar, or Langstroth hives. Of course, outside of beekeeping itself, there is incentive to practice conservation and implement specific land management strategies for pollinator health. And as Matus talked about, one species affects many others in the environment. And so in my research, a goal is to not only support beekeeping through the catching of swarms, but also long-term conservation of these species for their entire environments. Across Africa, the very large continent with many different unique biomes and ecosystems, there have been relatively few published literature, scientific literature on unmanaged honeybees. And free living colonies, the term that Honeybee Watch uses, can encompass, as Steve mentioned, colonies in nature-based beekeeping hives, in buildings, or in trees. But I'm specifically interested in those unmanaged honeybees that are away from urban areas often, or away from any sort of management, even if it's just a catch box in a relatively natural way with not too much interaction from humans. I'm interested in those colonies that are far away in natural areas that are the wild population supporting those other managed populations. So, of the limited research in Africa, very few of those surveys looking for those unmanaged colonies have been done by land area. And there's great value in locating colonies by land area, as we can monitor the changes in population density over time and get a little more of a pulse of how the population is doing. A lot of these studies also lack long-term monitoring. So we want to track nest sites and, if possible, with genetic tools, the colony survival itself over time to give more insight into how robust these wild populations are. I worked to characterize local nesting strategies of unmanaged honeybees in the Eastern Cape province of South Africa, and I did this by first describing the nest natural history features of bees in the area, as well as monitoring the seasonal changes of nest occupancy and damage. My study region is in the Eastern Cape of South Africa, so this is an incredibly biologically diverse area with over 4 different biomes meeting, and I had my home base in Makanda, South Africa, or formerly Grahamstown, which is home of Rhodes University, an important collaborator in my research. I established 5 field sites around Makanda that were a mix of rangeland, so could be used for livestock, but often you could picture a grassland or savanna, as well as game reserves. And these game reserves are your typical African safari, where you have natural areas stocked with megafauna for tourism. In both of these cases, I ensured there were no managed colonies within a 5-kilometer radius from the property boundary, and they're all at least 20 kilometers apart. So for some other genetic research that I'm doing, I have hopefully distinct subpopulations that on a typical foraging range, bees could not interact from one site with another. Before I dive into my exact methods, I'll first talk about the bees that I'm working with. There are 2 different subspecies of honeybee in South Africa. This includes Apis mellifera scutellata, which is the blue on this figure on the left. This subspecies has a range across southern Africa, and at least in the Americas, it has been made famous by the media of being a killer honeybee, known for its defensiveness. I could go on a whole soapbox of why defensiveness is not a good indicator of genetic lineage, but Apis mellifera scutellata does have that rap. And in green, at the very bottom of this figure, you can see confined to a smaller region in the more western Cape of South Africa is Apis mellifera capensis. And this subspecies is famous for the workers' ability to essentially clone themselves. So without mating with drones, they are able to produce viable eggs of other workers. Now, it's important to note that both of these subspecies are pretty much indistinguishable to the naked eye. They both have a lot of variation in color and size, and it requires molecular tools to tease them apart. I was working in the hybrid zone in between these two. So subspecies are us humans drawing lines in the sand, and if you are familiar with Harry Potter, this is more of a mudblood situation where there's a mixture. On the figure on the right is from a survey from managed colonies, so not those unmanaged or wild colonies, but of managed colonies. You can see the circle, that black area is the approximate study region I was in, and we have there almost half and half the blue and the orange for the 2 different subspecies. Now, both of these have smaller colonies than colonies of European descent and tend to abscond more frequently, which is a big thing to keep in mind as I talk about my research. So I have broken this project down into 2 parts. First, include my initial surveys to locate unmanaged colonies. I divided all of my field sites into square kilometer grids and semi-randomly chose grids to survey one at a time. I employed beelining techniques, which vaguely can be defined as following foragers from a nest from a resource, excuse me, to their nest entrance. In this case, I set up meter-high stands with sugar water mixed with anise essential oil as an attractant to recruit foragers. I used this mass recruitment strategy since I wanted to be as efficient as I could and also try to create a continuous highway of bees flying from my stand to the entrance that I could follow in the field. So from the stand, I would identify the direction that foragers were flying back, and then I would walk, stop, look, listen, and keep trying to follow that highway of bees in the sky back to their nest entrance. Normally, bees, when foraging, will meander from flower to flower, gradually getting enough nectar, but in this case, they're able to fill up their honey stomachs, and so when they leave that stand, they're making a beeline back to their nest. Here's a bird's eye view of this process. I have in a dashed line the outline of one of those survey grids, and those white squares represent a stand. I would set up at least 4 stands per grid, 1 per quadrant, to ensure we've thoroughly surveyed this land area. So we would follow all bee lines from one stand at a time, working strategically. We would, through the process of visiting all stands, saturate our survey coverage and have some sort of map like this of a mixture of colonies in the ground, in this case, or in trees. In total, we surveyed 16 square kilometers on foot and located 127 nests. We located 91 of these nests via beelining, and the rest were found by word of mouth or sometimes pure luck. The nest densities range from 0 to 13 nests per square kilometer, which tracks with some published research from Botswana. Now, interestingly, from the other talks you've heard today, Apis mellifera is generally described as tree-dwelling, but instead I found that a predominant nest type of my study area was colonies in the ground. So 69.3% of all colonies were in the ground, and these cavities were often dug by small mammals, aardvarks, mongooses, or warthogs, for example. We did find a quarter of colonies in plant cavities like tree trunks or limbs, but in the ecosystems in the study area, there just aren't a lot of large-bodied trees. I'll also note that I have relatively few colonies in rock faces and man-made structures like a roof or a wall. This is largely due to the design of my experiment. I avoided urban areas, so I don't have very many man-made structures, and I stayed away from rock faces. But other research from South Africa and the Western Cape has shown that unmanaged colonies do frequently nest on rock faces. So after locating this initial cohort of colonies, I then followed their nest occupancy over the course of 1 year at 3 different revisitation time points. Also want to point out the limitations of just studying the nest itself. So keep in mind that the bees in this area abscond frequently, so an empty nest does not necessarily mean that the colony died. The colony could have packed up, left, and reestablished in the surrounding area. Due to time and energy limitations, I was not able to re-beeline at each of these revisitations, so I don't have an exact idea of how many colonies are actually in that grid area at the time. In the same vein, an occupied nest is not necessarily the same colony. So in the time in between my revisitations, one colony could have left and moved out, and another could have moved in. So we're just going to be talking strictly about nest vacancy. At the end of my 12-month study period, a little over half of all nests were vacated. I confirmed this using an endoscope camera. So here you can see in the image on the left just the striations of wax where the combs were previously attached. I also documented any changes to the physical infrastructure of the nest itself, as predation is a big pressure in this area. So on the right, we can see an image of a termite mound, one of the frequent nest sites, and burrow dug by termite mound that has been dug out in between my revisitation time points. And here we're going to break down nest vacancy by my 2 most popular cavity types, so our ground nest and our plant nest. We can see over time that the overall nest vacancy increases, but that around 4 to 6 months of monitoring, or that second time point, we see a difference between the rate that the ground cavities are vacant compared to those nesting in trees. Now, this may be because ground nests are not as secure or more prone to predation or flooding events. This general trend matches what we can theorize about honeybee biology, that going into fall and winter, there are higher rates of nest vacancy with harsh conditions, but we see that stagnation of spring to summer, and that's likely due to swarming and reoccupancy. To conclude, ground nests were most predominant in this study area, which challenges the general conception about Apis mellifera. They may be the most abundant in the landscape or easy to locate, and this isn't the first time that honeybees have been confirmed nesting in animals of nests in other animals. This is just one example of how environmental events and land management decisions affecting other animals, in this case primary cavity nesters, could impact other secondary cavity nesters or other animals in the system, like honeybees. I could not do this work without all of my collaborators at Rhodes University and support from my home university at the University of Florida. So huge kudos to all of them. And if anyone would like to contact me directly, here's my email. Thank you so much.
You were perfectly on time. So thank you. Thank you to all of our speakers for both the information and being so much within the time frames. So, Kaylin, I love that you're expanding the knowledge on free-living colonies, especially in completely different geographies with different subspecies, mostly that we are not getting exposed to, because from what we've been doing with Honeybee Watch so far, the majority is in Europe. But super interesting, these nest ecologies and behaviors and all that. So thank you for sharing.